Hello, everyone. My name is Emma Collins, and I'm a student here at Our Lady of the Lake University in San Antonio, Texas. I wanted to say I'm super excited to be presenting here at Sigma Zeta this year, and I'm very honored for you guys to have me here. So thank you very much. I'm very excited. And today I'm going to talk to you about the fitness and phenological consequences of Populus trichocarpa and Populus balsamifera hybrids in a novel environment. Okay, so the first thing we want to discuss here is what exactly is phenology. So phenology is essentially understood as nature's calendar, or the timing of certain events through the seasons. So as organisms progress through the seasons, what they do is they reach a series of pivotal moments or milestones that actually dictate their life history events. So these can be broken down into milestones, and these milestones can be accelerated or delayed by biotic and abiotic factors. So things such as temperature, soil construction, soil nutrient availability, all those things can disrupt or accelerate or delay these milestones that these organisms reach during their lifetime. And disrupted milestones can actually influence overall lifetime fitness of these individuals. So if you are emerging too early, like if you're an insect, if you're emerging too early and you miss other ecological markers, that could be detrimental to your overall lifetime fitness. My project also has a little bit of hybridization involved in it. So some implications of hybridization here. Hybridization between populations acts as genetic shuffling. So when you have two species that interact and commingle and share their genetic material, you can actually increase the genetic diversity of their offspring. So when you have individuals that get together and they have babies, sometimes through sexual reproduction, they actually can mix their genetic material, and that increases the chances of adaptations in their offspring because of this new genetic makeup that they possess. Some objectives of the implications of hybridization are to characterize the role of hybridization in adaptation to climate. So we want to see how hybridization can affect these organisms' ability to adapt to a changing climate, and to inform predictions of future adaptation to changing climatic conditions and restoration efforts. So how are these organisms going to be able to adapt to changing climatic conditions, such as in the instances of global warming? And how are these hybrids going to perform if we use them in restoration efforts across the country? So really quick, we want to tell you where our trees are from. So these is a map of the tree origination points by transect. So you can see we have some up as far as Alaska, some from British Columbia, and even collected some out from Alberta, Canada. We collected 47 distinct genotypes. They were all rooted in Virginia at Virginia Tech and Jason Holliday's lab. And we acquired 2 replicate sets that were sent to each of the 18 common gardens across the United States. So each common garden that participated in this study received 2 replicate sets of the 47 genotypes. And here we are, sorry, I wanted to mention, here we are, San Antonio down there at the very bottom. So you can see that we're much, much further south than any of the origination points that these trees came from. And here, really quick, is just another map. These are all the common garden locations across the continental United States. So you can see there's quite a few up in the northern half of the United States, with a few being as southern as we are. And again, us in San Antonio, Texas, down there at the very bottom, we are the most southern location that was participating in this study. So methods. We employed a randomized block design. So that means we had 2 blocks that had random individuals randomly placed within them. And then what we wanted to do was record certain dates of phenology at 3 different stages that we observe in these trees. So the first stage we want to record was stage 2, which is bud flesh, which is when you can see a little bit of that leaf material there coming out of the bud. The second stage we wanted to record was stage 3, which is leaf emergence. And as you can see in the photograph there, that's when you see the emergence of a distinct petiole. So that little piece coming off the leaf there. And then the last stage that we recorded was stage 6, which is the bud set. So that's when we have this little capsule there at the top, which indicates that the tree is going to start becoming dormant for the season. We also recorded the height in centimeters at planting and at the end of the first year to estimate the growth rates overall. Oh, sorry, my picture's kind of in the way. Let me just move that a little bit. Okay. So another thing that we did was using an Aster model to analyze some of this data, which is an estimate of total lifetime fitness. And this accounts for dependencies between subsequent life history events. So that's important for something through like something with phenology, because we need these earlier stages to be incorporated so that we can generate this overall lifetime fitness. And this aster was used to estimate fitness for genotypes depending on the origin. So based on where you're from, we gave you a fitness for that. And here I have a graphic at the bottom. So we have planting, and then the first 3 nodes here, bud flush, leaf emergence, and bud set, were analyzed using a Bernoulli distribution, and then the final growth rate was analyzed using a Poisson distribution. Okay, and another quick note on methods here. All other analyses were done with generalized linear mixed models, such as the phenology analysis that we'll show you, a growth rate model that I'll show you, and all of our analyses were performed in R, specifically using the aster package and the Dharma package. Okay, so the first figure I want to detail with you guys is the data bud set. So we noticed that Alaskan trees actually took the longest to reach bud set. As you can see here, we're talking about maybe 85 days for the tree from leaf emergence to actually set its bud. And we think that this is because the phenology of the Alaskan trees was drawn out here down here in San Antonio, because San Antonio has an extended growing season compared to the growing season that's present in Alaska. So these trees are used to optimizing their growing season. So as soon as it's appropriate for them to release leaves, they're putting their leaves out and they're gathering as much sunlight as they can. And then at the latest possible moment, they are setting their buds that way that they've optimized basically the time that they can collect sunlight. And we think that it was drawn out in San Antonio because we do have an extended growing season down here because it is so warm. All right, I also want to discuss growth rate. So despite having the extended phenology that we noticed, Alaskan trees actually had the slowest growth rate. We think that this could have emphasis on resource storage. So we're thinking that while these trees have their leaves out longer, what they're doing is they're collecting as much sunlight and turning that into chemical energy as they can. And what they're doing is they're allocating those resources for when they need to use them through the long, harsh winters in Alaska. So instead of using that energy to grow taller like some of the other trees, they're actually conserving that energy and using it to persist through long winters so that they can survive instead of growing really tall in the summer and then dying in the winter time. We also think that these traits may be very plastic. So depending on where you are, depends on how you look is basically the way that you can talk about plasticity. So we're thinking that depending on the location that these trees are transplanted at, there could be a degree of plasticity in those traits. And here's our aster model that we made. So this is lifetime fitness by origin. So as you can see here, the fitness of the Alaskan specimens was actually determined to be the lowest. So they had a lifetime fitness that was significantly lower than any of the other transect sites, which was very interesting. And here I want to mention really quick the hybrid index. So we don't yet know how varying degrees of hybridization affects fitness. And we actually just received genomic data that's going to help us construct this hybrid index that will tell us what composition these hybrids have of each genotype and how that could possibly affect its overall lifetime fitness. And this one here, my picture is so in the way, sorry y'all. So this one details the distance of transects from Oulu, our San Antonio campus here. So this talks about the overall effective distance on lifetime fitness. As you can see again, the Alaskan trees have the lowest lifetime fitness, and they also have the greatest distance from Oulu. So we think that there are many components that likely contribute to this effect. Also really quick, I want to talk about my correlation coefficient here. So I have a R value of -0.86, which means that there's a pretty strong correlation to our best fit line, which was very interesting and very compelling. If we have strong correlation, that's usually a good sign. Yeah, but however, there are many components that likely contribute to this effect, such as change in latitude. So, as you change in latitude, you will experience a change in solar intensity. So when you're at the equator at 0 degrees latitude, the sunlight coming off from the sun actually hits the equator at pretty much a 90-degree angle. So you have high levels of solar intensity. As you move up and down the latitudes towards the poles, the light is diffused through the atmosphere, and it actually decreases in intensity as you move up and down those latitude lines. So as you can see here, the difference in latitude actually has also an effect on overall lifetime fitness. And again, these Alaskan trees are down there at the very bottom with the lowest lifetime fitness with the greatest difference in latitude. And we also think changes in latitude may also include several factors, such as differences in temperature. So as we notice here that there's the greatest difference in temperature was coupled with the lowest fitness in trees from Alaska. So all three of the figures kind of follow this pattern of the progression of trees down the best fit line, with Alaska always being with the lowest fitness. We think that this could have something to do with being locally adapted. And as you know, being locally adapted is excellent when you remain in that locality. So when you have adaptation specific to where you are grown and where you're from, especially as a tree, chances are you're going to do really well in that area because you've spent, years and years perfecting living in a certain area. So it's great for you when you're living in that area and you're locally adapted. And we also think a side note on that is that plasticity may be insufficient to overcome local genetic adaptations. So you might have some degree of plasticity, but if your genetic adaptations have greater effect over you, that plasticity is not going to be enough for you to be able to persist in an environment that's not where you're originally from. And that's another note we want to add there, is that narrowly adapted species will be hit harder by rapid climate change. So if you're specialized for a certain area and you perform really well in that certain area, that's what it means to be narrowly adapted. So you're only successful in a very specific environment. So you don't do well in a wide variety of conditions. So we think narrowly adapted species will be hit harder by rapid climate change, because if you are not used to varying climatic conditions and you're narrowly adapted to one, chances are you're not going to fare very well if you have a climate event and it's super hot and you are only used to living in the super cold. And some conclusions we want to draw here is that the greater the change in the environment, the more pronounced the negative effects on overall lifetime fitness. And our Alaskan trees pretty much displayed this perfectly. So we noticed they had the greatest change in latitude. They also had the greatest change in distance, and they also had the greatest change in temperature. And all of those had pronounced negative effects on their lifetime fitness. They constantly scored the lowest in terms of being overall lifetime fitness. They were always at the last in the race there. We also think dramatic environmental changes could pose deadly threats to locally adapted populations. So like I said, the more adapted you are to your locality, the better you do there. But if you are put into an environment that's not suitable to that, or climate conditions change rapidly and drastically, you're not going to do very well because you're so used to persisting in a specific kind of environment. And when it comes to replenishing biodiversity and damaged ecosystems, local species are the best option for replanting. So when you have species that are narrowly adapted and locally adapted, they're going to do well in where they're from. So when you're looking at replenishing damaged ecosystems, or replanting in areas where you've had, say, a lot of agriculture there and you need to replant trees to protect soil from erosion, or you had a natural disaster and you want to introduce some species back to protect the soil and stuff like that, you want to look for local species that are going to perform well in that area, that maybe are originally from that area, and that will help you with easy planting and maintaining those species, and they'll do great, just fine in that area because that's where they're from. And our next step in this project is constructing that hybrid index, or finishing constructing the hybrid index, because we really want to see what genetic combinations are allowing these hybrids to do well, and like where in these areas are the hybrids performing the best, and how the genetic factor plays into the overall lifetime fitness of these organisms. And some quick acknowledgements here. I just wanted to say a few thank yous. I want to say thanks to Dr. Mason Kulbaba, who is my mentor here at Our Lady of the Lake University. He actually introduced me to this project, and I helped when we first got it started. So I wanted to say thank you for inviting me on this project. It's been a wonderful learning experience. I also want to say thanks to Dr. Jill Hamilton and Dr. Jason Holliday, who have been integral in the beginning and got us off to a good start on this project, especially Dr. Holliday. He kept the trees very nice for us, and we were excited to have them from his lab, and they looked wonderful when we got them. I also want to thank the Sisters of Divine Providence for letting us plant on the convent grounds over there. They allowed us to plant our trees in a nice little quiet field so they wouldn't be disturbed. I also want to thank Getsemani Artiega, who's a good friend of mine, who's also working on a project with Dr. Kulbaba this semester, and she's been super at encouraging me and helping me out through this. I also want to thank Nancy McFerrin, who's our lab manager here at Our Lady of the Lake University. She was great at helping us get any of the materials that we needed for planting, and she's just always asking what she can do to help us get this project going. And I also want to thank Carter Guffey, who is my fiance, who also actually helped us plant some of the trees when we first got them from Dr. Holliday's lab. And he's just been a great encouragement and had me confident about this project and presenting. So thank you to everyone who helped me. Again, I'm so glad to present with you guys. And I'm excited to see everyone else's projects. All right, thank you very much.